Good morning students, myself Adhshya G R, lecturer in Anova College of Pharmacy. Today we are going to see about Anova, which means analysis of variance. Anova means analysis of variance. It measures two sources of variation in data and compares their relative sizes. Variation between two groups. For each data value, look at difference between its group mean and overall mean. It may be denoted by xi bar minus x bar the whole square. Variation within groups. For each data value, look at the difference between that value and mean of its group. It is represented by xij minus sij I bar the whole square. So Anova means it measures the sources of variation in data and compares their relative sizes. It measures sources of variation in data and compares their relative sizes. It is of two types: variation between groups and variation within groups. Between group mean is difference between group mean and overall mean. Within group means difference between the value and mean of its own group. Examples of obvious confounding. First, a clinical study in patients that allowed to take various concomitant drugs other than test drugs that affect the condition being treated. So, while clinical studies, patients are allowed to take various drugs other than test drugs. Except the test drugs, other drugs while taking these conditions will be treated by using Anova. Second, comparison of new tablet formulation to former formulation for dissolution with the tablets prepared by two different presses, one formulation on each press. Comparison of new tablet formulation. Difference in performance of presses can contribute difference in performance of presses can contribute to the difference in dissolution in addition to differences due to formulation changes. So the performance presses difference can be contributed to difference in dissolution due to the difference in formulation changes. What does Anova does? Consider an experiment to assess the effect of lubricating agent, dissipating agent and dissolution of the tablet. It considers while an experimentation, it considers the effects of lubricating agent, disintegrating agent and dissolution of tablets. The final analysis of variance would separate effects of these factors by computing the part of total variation attributable to lubricating and disintegrating agents isolated from the variation due to experimental error. This separation serves as a basis for testing a statistical hypothesis. Conditions used. In two sample situations, in paired setup, in repeated measures, when subject is measured at different times of points like 5 minutes, 15 minutes, 30 minutes and 60 minutes. Removing effect of covariate and regression. The Anova statistics is the ratio between group of variation divided by within group variation. F is equal to average variation between the groups divided by average variation within the groups. It is denoted by MSG divided by MSE. MSG is the estimated average variation between group means and MSE is the estimated average variation within the groups. So, and it is the statistical ratio between the group variation divided by within group variation. It is the average variation between the groups divided by the average variation within the groups. If the computed F ratio is less than 1, mean is non-statically different. If it is greater than 1, it is determinate. A large F is evidence against H0 since it indicates there are more different groups than within groups. Different types of ANOVA. ANOVA may be one-way ANOVA, two-way ANOVA, and mixed design ANOVAs. One-way ANOVA. Only one factor with more than two levels. One-way ANOVA has only one factor. Example, change of blood pressure in preclinical study and comparing two drugs under control. So, in one-way ANOVA, only one factor is used. Two-way analysis of healing. Two-way model is an extension to pair t-test. Two-way analysis of variance. It is also known as randomized block. Two-way model is an extension of paired t-test which is more than two groups of treatments are combined. Example, stabilities of 5 batches of tablet using 3 kinds of packaging material. Mixed design. Some factors are independent and others related. Application of ANOVA. A pharma company has 3 different medications for treating ulcers. By using ANOVA, we can determine the effectiveness by treating them. We can compare whether the medication works for better treatment and can choose the best one. In pharma company, the quality assurance officers collects 4 samples of A, B and C. Each tablet serves 6 tablets and measure the hardness of tablet. Means are significantly different from each other. Internal plot behaves means different between each other. Evaluation of bioequivalent studies. Second one is chi-square test. It is mostly non-parametric statistics used when data is represented in frequency or proportions. So chi-square test is a non-parametric statistics is used when data is represented in frequencies or proportions. Useful in discrete data. It is useful in discrete data. Any continuous data that can be converted into categorical data and statistics can be used. Continuous data which is converted to categorical data and statistics can be used. The chi-square test statistics can be used to evaluate whether there is an association between the rows and columns in continuous tables. More specifically, the statistics can be used to determine where there is any difference between study groups of proportions or risk factor internet. Chi-square test is the logic of hypothesis testing developed by Carl Pearson. It is used to determine any difference between the study groups of proportion of risk factors of interest. Is expressed by x square equal to summation of observed value minus expected value the whole square to expected value. The p-value where the calculated c-square is more than 0.1 at the confidence limit, 95% of it's not significant. Alternate formula for calculating chi-square value can also calculated by x square equal to n of ad minus eb whole square by a plus b plus b plus d plus a plus c into a plus c. Student t-test. Pharmaceutical company, they manufacture lakhs of tablets per day. In such cases, it is difficult to know the population parameters like mean, standard deviation. So we can apply test sample t-test. Types of t-test. There are various types of t-test, namely one sample t-test, two sample t-test, and paired sample t-test. One sample t-test. Here we use only one sample test when we have one sample group. Now in this test we use only one sample t-sets. By doing so, we would be able to find whether mean is significantly different or not regards with our target specification. For example, in a pharmaceutical company, quality assurance officers collect samples of 12 tablets to measure the dissolution rate of drug. Here we can find whether the mean of dissolution rate is significant to our target dissolution or not. Test null hypothesis, alternative hypothesis. So one sample test, we have only one sample group which is able to find whether the mean is significantly different or not with regard to the target specification. It can give the dissolution rate by individual value path of dissolution rate. Since our p-value is 0.019, which is less than our alpha value 0.05, we can conclude the mean value of dissolution rate is significantly different and the target value, target mean value, x bar sample mean value. Next one, two sample test. We use two sample t-tests for two independent sample groups. By doing so, you would be able to find whether mean is significantly different or not, and we would be able to compare one is good. For example, in an analytical qualification of two samples A and B, and each has 12 tablets to measure dissolution rate of the drug, which can compare which of the types have better dissolution rate, and we should be able to choose the best one. So to sample t-test, we take two independent sample groups. By doing so, we will be able to find whether means are significantly different or not. We will be able to compare with one which one is good or not. It is the graph for two sample t-test. Here we can conclude that mean of the dissolution rate is significantly different, but there is difference in variance or entity value. T-test. We use sample t-test when we have two different independent sample data of subjects. It is used to measure the effect of particular group before or after treatment. For example, during a clinical trial, we choose 20 patients and measure their weight before the drug treatment. And again, after 2 months, we again measure their weight. By doing so, whether the drug is effective or not. Here, there will be two sets of data. This is also called as dependent sample t-test. Yes, the patient weight is dependent on the drug treatment. Since our p-value is 0.007, which is less than our alpha value 0.05, we can conclude there is a significant difference between their treatments. We can visualize their difference by whether the drug is effective or not based on their weights. So, a paired sample t-test will measure value based on their weights. Thank you.